This is the Chapter 9 Proteins, Part 2 commentary. In this commentary, we're going to touch upon various ways to classify protein, both the overall protein itself and different levels of the protein based on its what we call primary, secondary, tertiary, or quaternary structure. We also talk about how to disrupt proteins, but let's not get ahead of ourselves. We'll start off with protein classification. In general, we like to classify proteins, make it easier on ourselves. This is one way we can classify them. Are they overall fibrous or are they globular? So for example, if I give you a protein that has this overall shape here, you can tell that it's roughly globular or spherical shaped. See how it's roughly in a sphere. So we call this a globular protein. Fibrous proteins tend to be long and rod-shaped or string-shaped. So in the next slide right here, this could be classified as a fibrous protein. This one down here would be classified as a globular protein. So make sure you can classify proteins based on its overall shape. Fibrous proteins tend to be related to structure, such as collagen or keratin, and are highly water insoluble. Globular proteins, they have diverse functions, such as transport proteins. This is the common shape that you find enzymes in, as well as your immune system, immunoglobulin proteins. So let's go to the next one. We also have another way of classifying, and we call these simple proteins versus conjugated proteins. Simple proteins have only amino acids present in the protein, whereas conjugated proteins have amino acids, and they also have some other organic or inorganic component. If it's an inorganic, such as a metal present, we call these specifically metalloproteins. If they have a fat or lipid present, we call these lipoproteins, and if there is a carbohydrate present, then we call these glycoproteins. I could give you a picture, and I'll make it really obvious whether it's a metal, lipid, or or a carbohydrate, and you'd have to let me know if it's at a metallo, lipo, or glycoprotein. The metal, lipo, or the metal, lipid, or carbohydrate component, oftentimes we refer to these as prosthetic groups. So just as a person who has prosthetic fingers helps the disability person pick up items, the metal, lipid, or carbohydrate prosthetically helps enzymes to be able to catalyze properly. So we call those prosthetic groups. This right here is an example of a prosthetic group, aka this is a glycoprotein. Glycoproteins contain carbohydrates along with their amino acid, along with their amino acid residues that make it up. It's hard to tell right here that this is a carbohydrate. I'll make it really obvious on an exam, for example, that this is a carbohydrate. But that is a glycoprotein. These are all under the general category, again, conjugated proteins. Now, if I were to sit there and take you to Google, here we see, if we go to a map software, that at this level of zoom, we're seeing individual countries and continents. if we were to zoom in, we could start to see states and cities. If we continue to zoom in, then we'll start to see freeways, and continued zooming would result in us seeing residential streets. So, my point in this is if we go back to the PowerPoint slides, the 4 levels of structure, you can think of these as levels of zoom when we're examining and understanding proteins. If we were to sit there and zoom out all the way, we would see quaternary and tertiary structure. Now, these are closely related. Quaternary structure really refers to proteins which are made up of even smaller proteins. The best example, and I'm going to go forward several slides just to show you. The best example of this is hemoglobin, where each one of the individual colors represents an individual protein that are all linked together via side chain intermolecular forces. So let's go back several slides. Tertiary structure is when we're looking at an individual protein, and again, it's due to its overall shape is due to side chain intermolecular forces. A little bit messy, so again, side chain intermolecular forces. Both of their structures, tertiary or if it's a multi-subunit protein, again, side chain intermolecular forces. It's like I'm going to test you on it. If we were to zoom in, so we're going to go ahead and zoom in, we could start to look at local protein properties. So the local protein properties or local structure within a single protein is often present, and we often see 2 types of this local structure. We see local alpha helixes and or local beta sheets. So in this example right up here to the top, in red what I'm circling, this green helical pattern is called a alpha helix. This is an example of secondary structure when we zoom into that level. If we take a look at the picture down below, the protein is mostly made up again of this alpha helix structure. All of it classified as secondary structure. The other type of structure that we're going to focus on is this internal ribbon-like pattern. We call these beta sheets. And again, this is another type of local secondary structure that we often see. Now, in secondary structure, we often focus on the fact that for secondary structure, it's due to backbone hydrogen bonding. Backbone hydrogen bonding. So this is evident in both alpha helix and beta sheets where present. Let me go ahead and focus a little bit more on this concept. I went ahead and I created a temporary slide right here. So in this example right here, we have a alpha helix, and what I've done is I've blown it up so that way in black you see the backbone of the polypeptide chain, or if it's greater than 15 amino acid residues, the protein chain. So this is the backbone. Now, on the backbone, nitrogen and carbons, we see, on the nitrogen, we see a hydrogen. On the carbon group, we see a carbonyl, and we see an oxygen with, hey, look, it has lone pairs. It turns out that on the alpha helical structure, the alpha helix structure is maintained and due to backbone hydrogen bonding. So if I were to sit there and draw, these two are too close to each other. If I were to sit there and draw a dash hydrogen bond between this hydrogen and this lone pair on this oxygen right here, this is an example of backbone hydrogen bonding. On the structure, basically we would see on a nitrogen, we'd see the hydrogen that comes out like this. On the carbonyl group, we'd see the oxygen. And again, we have the backbone hydrogen bonding. Now, although side chains are present, and I made these in red, the side chains are coming out like this, and because I wanted to, I just made them all leucines. These side chains have nothing to do with secondary structure. They're only responsible for tertiary structure, the overall shape of the protein. But when it comes to secondary structure, the only thing you need to look at is the backbone hydrogen bonding. So I've beat that to death. Again, backbone hydrogen bonding. We have two types where backbone hydrogen bonding creates secondary structure. The first type of secondary structure is alpha helix. The second type is beta pleated sheet. Here is a picture of an alpha helix in more detail. Notice on the right, the blue-green shows the backbone carbonyl groups and the hydrogens on the nitrogen groups forming backbone hydrogen bonding. So that's one secondary structure. Here's an example of beta pleated sheet. Notice these flat ribbons, again, are due to backbone hydrogen bonding. The very last one is primary structure. So if we were to zoom in all the way, we could focus on the overall sequence, and that's hard to read. So in this case, here we see the three-letter symbols. Primary structure was simply looking at the sequence of the amino acids. And based on the sequence of the amino acids, it determines what type of backbone hydrogen bonding will occur. For secondary structure, it also determines what type of potential side chain intermolecular forces, such as these two cysteines. What type of side chain intermolecular forces will be present. So the primary sequence helps determine what the secondary and tertiary structure is going to be. So the primary determines the secondary and tertiary, and in turn, if it's a hemoglobin molecule, for example, it will determine the quaternary structure. So I've beat those to death. You can go ahead and go through those slides for more detail. I do want to point out for secondary structure, oftentimes, if there's no structure that we can see locally, we just say that the chains are occurring randomly in random orientation. So technically, there's a third one for secondary structure, random chains, although I'll never test you on that one. So we're going to zoom ahead a bit since I've already talked about these. If we want to destroy, so if we want to destroy proteins, there's two different ways we can do it. One way is by protein hydrolysis, and that's where we use enzymes to basically chop up the protein into smaller polypeptides and to finally individual amino acids. So for example, when you're eating that drumstick of chicken, you're breaking up the protein into smaller polypeptides and then finally into amino acids, which are then transported into your body. The other way to break up proteins is protein denaturation. Basically what's happening is we're not chopping up the protein itself, we're simply disrupting the side chain intermolecular forces. So that way it becomes really loose with no actual tertiary structure. So we disrupt the side chains. Misspelled side chains, but we disrupt the side chains. That's an ugly, it's very poorly written. Let me write it again. We disrupt the side chains. So protein denaturation is when we disrupt the side chains. The other term for this, other than denaturation, is melting. So biochemists use the term melting, and that means when you disrupt its overall tertiary shape, forming no shape, and it just wags around like a loose noodle. How we do this is either by heating, or we can go ahead and change other environments such as the pH. We can change the salt, or we can change the solvent. We can add in an organic solvent. It's very easy to disrupt the side chain intermolecular forces, denaturing it. So make sure you can distinguish between protein hydrolysis and protein denaturation. Some other fine points on protein hydrolysis. Hydrolysis means it requires water in order to break apart the peptide bonds. So you use water to break apart, so therefore hydrolysis. If we wanted to not use enzymes and just use traditional laboratory methods, you can always use a combination of heat and an acid, or heat and a base. So you want to use both of them. If you just use heat by itself, then if you use heat only, then it will simply just make the protein fall apart. It will denature it, but it won't actually destroy the peptide bonds. So you need a combination of heat and acid for protein hydrolysis. So in this case, if I were to sit there and just simply heat it up only, I can disrupt it. This is an actual simulation of a protein where when it was heated, it now falls apart. Think of temperature as movement or vibration. So when we heat up a protein, we're basically causing it to over-vibrate. And basically the side chain intermolecular forces are overcome by the vibration, disrupting it. Therefore, it loses its overall shape, and therefore it loses its overall function. This is what usually happens in a first-degree burn. The cells may be okay at first, but all the proteins inside the cell fall apart, and since enzymes are made up of proteins, then the cell eventually dies. Good times. In this case, if you were to sit there and eat egg white is made up of nothing but proteins, or mostly proteins, and when you heat it up, it actually disrupts it and actually makes it really string-like. Consequently, these long strings of previous proteins will start to wrap around each other. And when they start to wrap around each other and clump, that's when you see this nice white solid. I do have a quick flash video of this if you're curious, also on the website. When it comes to denaturing proteins, there are generally 4 ways to do it. You need to memorize these 4 ways. Heat and ultraviolet light, organic solvents such as ethanol, hexane, or any type of organic solvent, strong acids or bases, and detergents such as soap. These are how they disrupt the proteins, so make sure you know these four methods and how they disrupt the proteins. Um, so heat and both ultraviolet light, again, they cause the molecules to vibrate. Ultraviolet light actually causes the bonds themselves to vibrate, and it disrupts the hydrogen bonds, ionic forces, denaturing the protein. Organic solvents, keep in mind we talked about the hydrophobic effect. So here's my protein right here, and then you have water which pushes in the protein, so that way you can avoid the nonpolar inside. Well, if you add an organic solvent, then that means there's no more water. Consequently, with a nonpolar organic solvent, then the hydrophobic effect is lost. Matter of fact, the protein may turn inside and out, so that way the nonpolar side chains is actually facing outwards. Pretty cool, huh? Strong acids and bases, it will interfere with the hydrogen bonds and ionic forces, which again causes it to fall apart. On the strong acids and bases, with prolonged exposure, it will eventually disrupt the peptide bonds as well. Although, when I ask you questions on these, they're only for denaturing proteins. And finally, detergents, soap. Soap is made up of, again, positive and negative charges, which will compete with and interfere with the side chain intermolecular forces.